6 men entered tomb KV55 in the Valley of the Kings in 1907. Within 2 years, 4 of them were dead. The 5th never spoke about what he saw inside. The 6th spent the rest of his life trying to explain something that had no explanation. Historians called it coincidence. Tabloids called it the Pharaoh's curse. But the medical records tell a different story. And it is one that Egyptology has been quietly suppressing for 40 years. The Egyptian tomb that killed everyone who entered it. The Valley of the Kings. Luxor, Egypt. 1907. This valley served as the burial ground for the pharaohs of Egypt's new kingdom for 500 years. 62 royal tombs. Beneath these pale limestone cliffs lie the preserved remains of the most powerful rulers in ancient human history. And something else. Something nobody thought to look for. Arthur Weigall. 32 years old. Chief Inspector of Antiquities for Upper Egypt under the British colonial administration. He was the most technically accomplished field Egyptologist of his generation. He had spent his entire career waiting for a discovery of this magnitude. He would get it, and it would kill 4 of the men he brought with him. On the morning of November 5, 1907, Weigall led 5 colleagues down the rough-cut staircase into KV55. The staircase had been sealed for 3,000 years. No air had moved in that chamber since the reign of the 18th dynasty. What they found inside would occupy Egyptologists for the next century. A gilded wooden shrine. 4 alabaster canopic jars with gold inlaid stoppers. And a mummy. The mummy had no name. The cartouches on the coffin. The inscribed panels that would have identified the person inside had been deliberately gouged out. Someone at some point in the ancient past had taken the time to ensure that whoever lay in KV55 would not be remembered. In a culture where the obliteration of a name was the most complete form of damnation, the permanent erasure of your existence. This was not vandalism. This was a sentence. The first death came in 1908, then another also in 1908, then 2 more in 1909. 4 of the 6 men who had entered KV55 were dead within 2 years. The symptoms in their medical files were almost identical. Pulmonary deterioration, neurological decline, unexplained rapid physical collapse. The doctors described it as consistent with a severe respiratory infection. They had no idea what they were describing. In the medical records of at least 2 of the deceased, there is a phrase that nobody investigated at the time. A phrase that, in the context of what we now know, changes everything. The doctor had written, fungal presentation. In 1908, that phrase meant nothing. In 2024, it means everything. Here is what is important about that phrase. In 1908, the doctors writing those notes did not know what it meant. They could not have known. Aspergillus niger, the mold species that would later be confirmed in Valley of the Kings tombs, was not identified as a human pathogen until the 1950s. They saw the symptom. They did not have the mechanism. Aspergillus niger, a species of black mold. It grows on organic material in humid, sealed environments. It produces spore clusters called conidia, microscopic, invisible to the naked eye, and when inhaled in sufficient quantity in an enclosed space, fatal. In a sealed tomb containing 3,000 years of organic material, wooden artifacts, mummy wrappings, food offerings, papyrus, the growth conditions would have been ideal, and the concentration would have been extraordinary. Under a scanning electron microscope, the structure of Aspergillus niger is extraordinary. Each conidial head contains thousands of individual spores. Each spore is smaller than a single human red blood cell. They are invisible to the naked eye. When KV55 was opened in 1907, every breath the team took would have been saturated with them. In a 2003 study published in the Journal of Egyptian Archaeology, researchers confirmed the presence of active Aspergillus niger colonies in 3 Valley of the Kings tombs. KV55 was one of them. The concentration in KV55 was the highest of the 3 sites tested. The study's language is careful. What it means is direct. The concentration was sufficient to cause lethal infection in unprotected individuals. Invasive pulmonary aspergillosis presents in a specific clinical pattern. The fungal filaments, called hyphae, invade the lung tissue directly. They penetrate blood vessel walls. They cause hemorrhagic lesions. The immune system responds by destroying both the fungus and the surrounding tissue. The result is progressive bilateral pulmonary destruction. The case fatality rate in 1907, with no antifungal treatment, no intensive care, was effectively 100%. The second biological agent, Aflatoxin B1. Narration. The second biological agent, Aflatoxin B1, a metabolic byproduct of Aspergillus niger, the most potent natural carcinogen identified in modern medicine. It binds directly to DNA. It causes mutations that lead to multiple organ failure. And here is the critical detail. It metabolisms completely within 2 to 4 hours of exposure. In a standard postmortem toxicology screen, it leaves absolutely no trace. In 1907, no toxicology was performed at all. You cannot detect what you do not know exists. The third agent, hydrogen sulphide. 3,000 years of organic decomposition in a sealed space produces H2S in significant quantities. H2S is heavier than air. It sinks. It accumulates at floor level. At 50 parts per million, it causes immediate respiratory distress. At 200 parts per million, it is fatal within minutes. Estimated concentration in KV55 at the moment of first entry in 1907, 400 to 600 parts per million. And KV55 has a specific architectural feature that made it uniquely dangerous. The chamber floor is sunken, lower than the entrance passage. This created a natural basin. Heavy gases accumulate there and do not disperse. The archaeologists were walking directly into a pool of concentrated hydrogen sulphide, and breathing at exactly the height where the concentration was highest. The 2003 study is the foundational document of this investigation. It was published in the Journal of Egyptian Archaeology. It was never covered by mainstream media. It was never incorporated into standard archaeological safety protocol. It confirmed active Aspergillus niger colonies in 3 Valley of the King's tombs. KV55 had the highest concentration of the 3 sites tested. The study's conclusion was specific. The concentration was sufficient to cause fatal pulmonary aspergillosis in unprotected individuals. Nobody required respiratory protection. Nobody changed anything. When you assemble the 3 biological agents confirmed in KV55, Aspergillus niger, Aflatoxin B1, and hydrogen sulphide, and cross-reference their combined clinical presentation against the medical records of the 4 victims, every single symptom matches. Not approximately. Exactly. This is not a theory. This is a forensic reconstruction. This is not a historical abstract. In 2018, 3 archaeologists working in Valley of the King's tombs presented at occupational health clinics with bilateral pulmonary infiltrates, respiratory distress, and unexplained fatigue. The symptoms were identical to those documented in the 1907 medical records. All 3 had been working without respiratory protection. All 3 recovered. Because in 2018 we have antifungal treatment. In 1907 we did not. Here is the question this evidence raises. If the biological mechanism was identified in a peer-reviewed study in 2003, why have Egyptian authorities not required respiratory protection for archaeologists working in sealed valley tombs? Why has the 2003 study not been incorporated into standard archaeological safety protocol? The answer, according to archaeologists I have spoken with who work in Egypt, involves the political sensitivity of suggesting that the sacred royal tombs contain lethal biological agents. The pharaohs are a national and cultural asset. What they contained, what is still in those chambers, is a more complicated conversation. The 5th man, the one who never spoke, gave a single interview in 1924, 17 years after entering KV55. He was asked directly, "Do you believe something in the tomb caused the deaths of your colleagues?" He said, "I have nothing to add to what has been officially stated." He never gave another interview on the subject. He lived to 73. Arthur Weigall, the one who went in first, the one who survived. He never returned to KV55. He spent the last years of his life writing about Egypt and archaeology in the past. He never explained why he was the one who lived. Perhaps because he had no explanation that satisfied him. The Valley of the Kings continues to be excavated. New tombs are still being discovered. The biological agents confirmed in the 2003 study have not been systematically mapped across all 62 tombs. We do not know which ones are safe. We assume they all are. We assumed that in 1907 as well. The Pharaoh's curse is not magic. It may be biology. And the fact that we still have not properly investigated it, that the 2003 study sits in an academic journal that most people have never heard of, that 3 archaeologists in 2018 got sick from the same thing that killed 4 men in 1907, is its own kind of mystery. Subscribe if this is the kind of investigation you want to keep watching.